Hello again. So we left off, I believe, on this slide in class today, talking about the difference between plasma cells and memory cells. We're going to come back to plasma cells and memory cells in a few minutes, but first, we're going to talk about antibodies. Excuse me. And if you remember, B cells attack antigens in the blood, or pathogens in the blood, okay, by producing antibodies that are specific to specific antigens. Now the question is, what do antibodies do to the antigen or the pathogen that's coming into the body? Again, just to make sure the difference between pathogen and antigen. Pathogen is like the entire bacteria or the entire virus. So this is the entire bacteria or the entire virus. The antigen is the part of the bacteria or virus that the antibodies recognize or can bind to. So the next part of this lesson is going to focus on antibodies, their structure, and what they do to help sort of tag or mark antigens in the blood. So, antibodies are proteins that bind to specific antigens in the blood. They're composed of 4 chains. We have a heavy chain and a light chain, and then a heavy chain and a light chain on the other side. Now, there are 2 major parts of an antibody. The 2 main parts are the variable region, which is shown here in V, and then the constant region, which is shown here marked with C, and it's purple. It is kind of like it sounds. The variable region is different on every antibody, and the constant region is the same. This part of the constant region over here, that's the part that inserts in the cell membrane of the B cell. The variable region is the region that binds to the antigen. In some ways, the variable region is kind of like an enzyme. It has a specific molecular shape that matches and can bind to the antigen. Each antibody that B cells make is unique and specific. One antibody can respond to one antigen, and there are millions of different antibodies that can be made by B cells. In some ways, and we're going to look at this in a little bit more detail in a second, antibodies are kind of like handcuffs. They bind to the antigen and tag it, being like, I've got you. You're now going to go to jail. They tag foreign particles or antigens that they recognize and say, you are not supposed to be here. I am going to handcuff you until help can come and get rid of you. Now, once again, too far. The structure of antibodies is important, as I continuously go too far. So again, we have the variable region, which in this case is in yellow. And then we have the constant region, which is the same in most antibodies. Notice how the constant region, or at least part of the constant region, is inserted into the cell membrane of the B cell. Now, also important, you can see in the structure of an antibody, it's a protein, and you can see these atoms in this case, and those represent disulfide bridges. Disulfide bridges hold together the tertiary structure of an antibody. Don't worry so much about the difference between light chains and heavy chains. We really care that you know more about the difference between the variable region and the constant region. Now, the question is, and we're going to look at it a little bit further, is what do antibodies do once they recognize and bind to antigens? So, there are a couple things that they do. Okay? And again, this is a very complicated drawing, which we're not going to go through all the parts. But, essentially, what antibodies do is they bind, in all cases, they're binding to the antigen that they recognize that is on the foreign particle. So here we have a virus, and we have many antibodies binding to the virus. This virus, at this point, cannot infect cells. If you remember, viruses infect cells by attaching to receptors. At this point, the virus is neutralized. It cannot infect cells, as its protein coat is, being occupied by antibodies. Same thing here with the bacterium. This bacterium is neutralized. It cannot go in and infect cells. Antibodies also can cause bacteria to group together or agglutinate them so that they cannot infect cells. We are not going to talk about these two parts of the immune system as they're not on the AP exam, and we do not have time to discuss them. Anyway, the point is that in both of these cases, the antibody acts like a tag, saying that this antigen is foreign. We need to get rid of it. And the binding, this is the most important part, the binding of an antibody to an antigen causes macrophages, which we talked about in the beginning of this lesson today, to recognize these particles as foreign and engulf them and then digest them in their lysosome. That's how macrophages know which particles are foreign. They are tagged by antibodies that have a specific binding region that recognizes them. Now, you are born with millions of different antibodies coded for in your DNA. And like we said, these antibodies bind to foreign pathogens and mark them for destruction. So what that means is in your blood, there are B cells circulating in your blood, each which makes a different antibody. So there are millions of B cells in your blood that are floating around in your blood, and they make a specific antibody. Now, even with that number of B cells making different antibodies, that's still not, there's not enough possibilities in your DNA just naturally to be able to tag all foreign pathogens that are possible to be in your blood. We're not going to talk about this now, but eukaryotic organisms, particularly advanced animals with advanced immune systems, have evolved ways to shuffle genes to make almost an infinite number of antibody combinations. On Monday, we're going to talk a little bit more about how, particularly mammals, do this. How that's possible, that using something called gene shuffling, we can make an unlimited number of antibodies if needed. But for now, it's enough to know that our body naturally makes about 1 million different antibodies. And these B cells are circulating in the blood, each carrying their own antibody on their cell membrane. Okay, we're now going to look at the entire B cell response. There are 2 slides that follow. One is a huge picture, and the other one says B cell response in words. You can choose to ignore that slide and simply write what happens on the big picture, or you can use numbers and write what happens on the empty slide. It's up to you. So let's take a look at the B cell response. So in this picture here, we have a foreign invader, and on the in this case it looks like a bacterium, and on the bacterium there are these red particles that are the antigen. And this bacteria has gotten into your blood and is in your blood. By chance, a B cell, that has an antibody on its cell membrane that can bind to the antigen, binds to the antigen and recognizes the antigen. This activates the B cell. When the antibodies are the right shape or recognize the antigen, the B cell becomes activated. And again, this is happening in the blood. Now, the activation of the B cell causes it to clone itself. And what cloning means is that it causes it to divide rapidly, making many, many copies of this B cell, which before there weren't that many in the blood. Now there's thousands of this B cell in the blood. And these B cells, some of them become plasma cells and start producing huge amounts of the antibody that can bind to the foreign invader. Some of these B cells that have been produced become memory cells that aren't producing antibodies, but they stick around in the blood. And the purpose of the memory cells and why they're there is that if the infection comes again, maybe a year later, a month later, the immune cell response will be quicker the second time you're exposed to the same pathogen. Now, we have these antibodies that are being produced by these thousands of plasma B cells. These antibodies are released into the blood, and they float around in the blood, binding to all the bacteria that are displaying the antigen that was originally recognized. Now, once the antibody binds to the bacteria or the antigen on the bacteria, it signals, it's like it handcuffs it and signals for, hey, macrophages, or phagocytic white blood cells as we also call them, it signals for them to come and ingest, through phagocytosis or endocytosis, those words mean the same thing, the marked bacteria. And again, it's marked because the antibody can specifically bind to this antigen, and that handcuffs the bacteria and marks it for destruction by the macrophages. That was a lot. I would pause the video at this point or replay the parts that you need and either write on this slide here, or go back to the next slide and write in steps, number this diagram, and then write in steps what happens. Now you notice it takes, for a full response, it takes 10 to 17 days. And the question is, why does it take so long? And the answer is, well, it takes a long time, first of all. In the beginning, there aren't that many B cells that produce the antibody that can bind to this antigen. So it takes some time for the B cells, the few that there are in the body that produce this antibody, it takes a while for that B cell to find the foreign invader. It also takes time for once the antigen is recognized by the B cell, it takes time for the B cell to replicate and produce enough antibodies to be able to capture and mark for degradation all of the bacteria that's been gotten into your blood. So a full response, the first time you're exposed to an infection, takes about 10 to 17 days. Now, what are vaccinations? Vaccinations are an inactive form of the pathogen that has the antigen on it. It cannot make you sick, but it stimulates B cells to start producing plasma cells and memory cells. But it's an inactive, non-pathogenic, meaning it just can't kill you or hurt you, form of either the virus or bacteria, usually most vaccines are for viruses, that activates the immune system and causes the B cells, that B cell that can recognize it, to start producing memory B cells. As a result, if you get exposed to the real infection, you won't have to wait for all the replication to occur, and there'll be more B cells in the blood that will be able to respond to the antigen, so they'll find it quickly if the real pathogen enters your body. Again, it's important to realize that vaccines are the inactive form of the virus. We need to go into this a little bit further, and I'm running out of time because, again, my videos can only be 15 minutes long. So I'm going to upload this video and then come back for the last 2 slides. So stay tuned.